We will again discuss two core hallmarks. The hallmarks directly related to survival are resisting cell death and enabling replicative immortality. Let's start with resisting cell death. As you may know, there are several forms of cell death, of which some are programmed and planned and others are not. Apoptosis is one form of cell death which is strictly organized and regulated and is useful to remove damaged or old cells and thus play an important role in tissue homeostasis. The cell shrinks, nuclear and cellular fragmentation occurs, and so-called apoptotic bodies are formed, which can be removed. Autophagy or self-degradation is another form of cell death which is usually programmed or at least strictly organized. Necrosis, on the other hand, may be seen after trauma or injury and is resulting in acute damage. During necrosis, the cell actually explodes and releases its cell content in the environment. What we see in cancer cells is that they usually undergo less apoptosis and autophagy, whereas necrosis is more common. Necrosis may also contribute to the process of cancer development, since the cell contents that are released may act as signals that induce, for example, inflammation or other cancer-promoting factors. An example that is relevant to resisting cell death is the famous tumor protein 53 or p53 in short. p53 is a protein that is able to see and screen whether there is damage in the cell. So p53 also stops the cell cycle in the G1 phase, so there is time to repair the DNA. If the DNA cannot be repaired anymore, p53 can also facilitate apoptosis. In cancer cells, p53 might be inactivated, and therefore the option for programmed cell death is ignored. The second core hallmark related to survival is enabling replicative immortality. In other words, the cells stay alive. I don't know if any of you have worked with a cancer cell line before in the lab, but there you can clearly see this immortality capacity of the cells. What you see over here on this microscopic picture is a specific cancer cell line in a petri dish. And under favorable conditions, the cells can just keep dividing. Another word for this replicative immortality is unlimited replication, and for this characteristics the telomeres are very important. As you may know, DNA in the nucleus of the cell is organized or folded in the forms of chromosomes. Telomeres are the DNA sequences that form the ends of the chromosomes. These end caps are shown here in orange. With each cell division, the telomeres becomes a bit shorter. At a specific moment, the telomeres are so short that the cell reaches a specific state which is called senescence. Cells are still alive and functioning, but the cell division stops. After being in the senescent state for a while, the cells enter another stage which is called crisis, and now the cells undergo apoptosis. This is a normal process and it is responsible for clearance of old cells and maintenance of a healthy tissue structure. In cancer cells, this process, however, may be disturbed, and let's see what happens. As a summary, in normal or healthy cells, the number of times that a cell can divide is limited. This is because the telomeres become shorter. Cells enter the senescent phase and ultimately undergo cell death. Cancer cells, however, are able to prevent shortening of the telomeres, and that is because they can express an enzyme, called telomerase, that is responsible for extending the telomeres. Therefore, the cancer cell prevents cell death and becomes immortalized. To wrap up, we've now seen 4 core hallmarks related to growth and survival. In clip 1, we have discussed chronic proliferation and we've seen the important role for growth factors in this hallmark. We've also discussed escaping or ignoring the stop signal, and we should be able to understand the role of tumor suppressor genes in here. In the second clip, we have discussed avoidance of cell death and the importance of apoptosis, necrosis, and p53 should be recognized. And also we have discussed unlimited replication or immortality, and here senescence and telomerase are important keywords.